Good afternoon, everyone. Thank you, Dr. Jones, for inviting us to present our work on interleukin-1 and interleukin-6 receptor modulators to prevent preterm birth. Preterm birth is a major cause of medical complications worldwide. It's a major cause of infant mortality, such that approximately 1.1 million children every year die out of the 13.5 million children that are born prematurely. In addition, it's a major cause of morbidity, notably that associated with neurodevelopment, but also in long-term predispositions to cardiovascular disorders and metabolic disorders such as diabetes. Accordingly, preterm birth remains a major medical cost problem in the United States, amounting to approximately $40 billion a year. Despite advances in neonatal care, preterm birth is a major unmet medical need. Preterm birth is actually triggered by a number of risk factors. But there is one major risk factor, which is a converging risk factor, and that is infection and inflammation. And this is based on a number of studies, clinical correlations, immunomic studies, proteomics, multiomics, microbiomic approaches that have actually demonstrated the important role of infection and inflammation as a major risk factor for preterm birth. Despite this evidence demonstrating an important role for uteroplacental inflammation in triggering preterm birth, the only medications that we currently have to tackle preterm birth is tocolytics. But these agents act very much downstream of the cascade resulting in preterm birth. Essentially, ascending infections and associated inflammation trigger toll-like receptors, which then actually lead to a production of chemokines and cytokines, including interleukin-1 and interleukin-6. These then in turn recruit and activate immune cells, which then actually lead to the production of uterine activating proteins essential for fetal expulsion. This leads to myometrial contractility, rupture of membranes, and fetal expulsion prematurely. But in addition, these inflammatory mediators have direct detrimental effects on the organs of the fetus, resulting in neurological, respiratory, and gastrointestinal complications. But right now, the only thing that we have to offer to these patients are tocolytics, which are acted downstream and hardly have any effect in prolonging gestation. Evidence for inflammation is paramount. In 2014, Roberto Romero collected amniotic fluid from women that went into labor and delivered prematurely. And what he showed is that the more the infants were premature, the higher actually the percentage of inflammatory markers present in the amniotic fluid. Among these inflammatory markers, interleukin-1 actually turned out to be quite an important one. It was high in concentrations. Interleukin-1 is known to amplify inflammation. Polymorphisms of human interleukin-1 or its receptor or the enzyme that produces it, caspase-1, is associated with longer gestation because it disrupts actually the interleukin-1 signaling. And interleukin-1 itself induces preterm birth in numerous animal models. Based on this evidence that interleukin-1 exerts a major contribution in resulting in the cascade of events leading to myometrial contractility, rupture of membranes, and preterm birth, it was therefore logical to propose that an interleukin-1 receptor antagonist acting upstream of the cascade of events would actually interfere with these events and prevent preterm birth, as well as preserve fetal organ integrity. A number of interleukin-1 receptor antagonists are currently available on the market. This includes Kineret, anakinra, canakinumab, which is an anti-IL-1 antibody, and the interleukin-1 inhibitor rilonacept. But these agents are large molecule biologics that have mitigated efficacy in preterm birth. In addition, they can actually cause serious infections. They have immunogenicity and are very costly therapeutics. Accordingly, we decided, therefore, to develop our own interleukin-1 receptor antagonist by actually designing a small peptide based on the concept that by reproducing a specific region of a protein of interest, one could actually use this protein to be able to inhibit the activity of that protein of interest. Interleukin-1 receptor is composed of three subunits. The interleukin-1 receptor accessory protein, from which actually our peptide, which is named Ridvela, arises from. It corresponds actually to the amino acid sequence of that region. The other subunit is interleukin-1 receptor type 1 and the interleukin-1 ligand itself, which interacts with both subunits. So Ridvela turns out to be an allosteric antagonist of the interleukin-1 receptor. It is specific, it is potent, it has an inhibitory constant of approximately 10 nanomolar. It's effective in numerous models of inflammation. It's stable because it's in a D conformation and therefore it is not prone to degradation by endogenous proteases. And no adverse effects have been detected so far. Ridvela mitigates preterm birth in a preventive as well as a treatment mode, and it also improves newborn outcome, and this was shown in 5 different species. Consistent with the region from which it actually arises and the area where it is postulated to interfere with the activity of the interleukin-1 receptor, Ridvela does not act as an orthosteric inhibitor, as is the case for the currently available interleukin-1 receptor antagonists such as canakinumab, Kineret, and rilonacept. Ridvela actually is able to interfere with some of the canonical pathways associated with interleukin-1 receptor activation, specifically the Rho kinase and the MAP kinase pathway, but it actually preserves the immune surveillance pathway of the NF-kappa B. Whereas Kineret, for instance, interferes with all the pathways and is associated with complications. We proceeded to evaluate the efficacy of antenatal Ridvela in preventing preterm birth. And we used 3 different models. And in all 3 cases, either induced by interleukin-1 or by gram-negative components, such as lipopolysaccharide, or gram-positive components, such as lipoteichoic acid, in all 3 cases, Ridvela was able actually to markedly inhibit prematurity associated with these triggers. Moreover, antenatal Ridvela was found actually to preserve organ structure and function in these mouse neonates. So we looked at, for example, brain mass, which was actually found to be markedly diminished by interleukin-1. In the presence of Ridvela, this is quite preserved. Brain function is totally preserved by Ridvela, and lung development, specifically alveolar count, which is markedly affected by interleukin-1, is actually totally preserved in the presence of Ridvela. In summary, when we compare Ridvela to Kineret or nifedipine, Ridvela by far outperforms Kineret in prolonging gestation, in enhancing survival of the fetus and newborn, and in preserving newborn brain, retina, lung, and gut integrity. This is also much better than nifedipine, which is essentially ineffective on all of these comparators. Moreover, it's important to point out that we did not see any adverse effects associated with Ridvela, whereas Kineret actually interferes with efferocytosis, which is really important in immune vigilance, and it also was found to cause fetal acidemia. This efficacy of rubella was confirmed by a number of separate laboratories and in numerous species as well. Yet, interleukin-1 is not the only mediator actually implicated in triggering preterm birth. IL-6 is also found in high concentrations in amniotic fluid. IL-6 stimulates uterine contractions by inducing oxytocin receptor expression in rats. The null mouse of interleukin-6 is refractory to low doses of lipopolysaccharide. Genetic deficiency in IL-6 delay parturition, and IL-6 receptor antibody is found to actually prevent preterm birth induced by inflammatory stimuli. But what does IL-6 inhibition do on fetal and newborn organs? Is it effective? In addition, would there be a preference to using allosteric modulators to provide safer pharmacoselectivity? And this is what we decided to investigate. The interleukin-6 receptor is a hexameric structure composed of two interleukin-6 receptor moieties, two GP130 moieties which interact with the interleukin-6 receptor, and two ligands of interleukin-6. These actually, when the process is activated, results in canonical signaling of MAP kinase, AKT, and STAT3. We therefore proceeded to use the same approach as for interleukin-1 receptor and other receptors in designing a peptide that would interfere with the actions of interleukin-6 receptor. And in this case, I'm showing you two different regions of the interleukin-6 receptor from which actually we designed peptides. You see 633 as well as HSJ 639. We tested actually the efficacy of these peptides on an IL-6 induced TNF-alpha expression in het blue cells. And you could see actually, which is highlighted in red, that 633 and 639 turned out to be quite effective in het blue cells containing the interleukin-6 receptor. In addition, 633 was also effective on human fetal membranes stimulated with HMGB1, which actually activates toll-like receptors, and this effect was actually inhibited by 633. However, in mouse uterus, 633 was found to be quite effective in reducing uterine activating proteins, but 639 was not reproducibly effective. So we therefore decided to pursue the rest of our study by focusing on 633 as an anti-IL-6 receptor modulator. We hereby show that actually 633, radiolabeled, binds to cells that actually contain the receptor, but not to cells that actually are devoid of the receptor. Moreover, this binding actually is displaced by the presence of cold 633. In addition, radiolabeled 633 was actually not displaced, however, by interleukin-6, suggesting therefore that 633 binds to IL-6 receptor in a region distinct from that of the ligand binding region of IL-6, and therefore is not an orthosteric inhibitor but acts more like an allosteric inhibitor. We therefore proceeded to study the efficacy of dose-dependent concentration of 633 in inhibiting interleukin-1 expression in cells stimulated with interleukin-6. And you can see in here that actually increasing concentrations of 633 do annul the efficacy of interleukin-6 with an IC50 of less than 1 nanomolar, 0.8 nanomolar. We further show that actually 633 interferes with some of the pathways associated with transduction of the interleukin-6 receptor, specifically the STAT3 pathway, but it actually preserves the MAP kinase and AKT canonical pathways, and these are very important in cytoprotection. We then moved on to study the efficacy of antenatal 633 in the prevention of preterm birth and neonatal injury. Antenatal 633 was found to prevent lipopolysaccharide-induced inflammatory surge in gestational tissues, in uterus, in placenta, and in fetal membranes. You could see that LPS actually induced interleukin-1 and interleukin-6, and this was actually totally abrogated in the presence of 633. Consistent with these effects on inflammation, 633 also was found to prolong gestation. As you could see in here, 633 normalized actually gestational length even in the presence of LPS to the same extent that an anti-IL-6 receptor antibody did. You could see also and appreciate that newborn survival was normalized by 633 to an extent similar to that seen with anti-IL-6 receptor antibody, which is a much larger molecule. In addition, 633 not only prolonged gestation to enhance maturation of the fetus, but it also actually had a direct effect in preserving integrity of tissues. And here we show actually the brain, where actually in the presence of LPS, there is a vasoattenuation, and this effect is actually preserved by 633. Similar benefits were also observed on the lungs and the gut. In order to assess whether 633 went across the placental compartment to act as an anti-inflammatory in the fetal compartment, we tagged 633 with FITC, a fluorescent tag. And what we can appreciate in here on this slide is that in the presence of FITC tagging 633 only, there is no crossing of the placenta. Whereas in the presence of LPS, FITC alone is able to cross a little bit, but when actually 633 is tagged with FITC, there is quite a bit of crossing across the placental into the fetal compartment, as we can appreciate on this intensity of fluorescence in the presence of FITC. In order to assess whether 633 acted via the soluble IL-6 receptor versus the membrane-bound IL-6 receptor, we pursued the following experiments. So in human amniotic epithelial cells, which are devoid of the membrane-bound IL-6 receptor, if you stimulate these cells with IL-6 in the presence of vehicle, there is no IL-6 receptor, so therefore you get no signal transduction, no increase in phosphostat 3. But in the presence of the soluble IL-6 receptor, then you actually do get an increase in phosphostat 3, as the soluble IL-6 receptor, once it gets activated by the IL-6, it actually comes to the membrane and combines with the GP130. In vivo, we pursued these experiments to see actually whether the soluble IL-6 receptor could contribute to the prematurity related to IL-6 and its inhibition by 633. And in fact, one can appreciate in here that what we did is we actually utilized a trap of the soluble IL-6 receptor, which is the soluble GP130. And in the presence of soluble GP130, we normalized actually LPS-induced prematurity to term, as one can appreciate in here on this violin graph, and similar to that seen with 633. Hence, what we can say is that IL-6 dependent LPS-induced preterm birth is largely attributed to the soluble IL-6 receptor. In summary, for Ridvela and 633 properties. Ridvela and 633 display unique properties, being pharmacologically selective by favoring bias signaling. That means it affects some but not all of the signal transduction pathways coupled to the respective receptors. Rivella has been shown in different studies to exert anti-inflammatory properties in distinct conditions. More relevantly, Rivella and 633 in blinded studies have been shown to diminish gestational tissue inflammation, to prolong gestation induced by inflammatory stimuli, the major causes associated with preterm birth, in preventive and therapeutic modes, to enhance pup survival and improve their outcome, to improve newborn outcome, including preservation of organ histology and function, by suppressing prolonged inflammation and resulting in normal growth trajectory. These in vitro and in vivo studies have been confirmed by separate laboratories in numerous species. Accordingly, Rivella and 633 are first-in-class therapeutic candidates for safe prevention and arrest of preterm birth. And with this, I thank you for your attention and will now take questions.